So if we keep going here, as we mentioned, this is just a theory, but it's pretty commonly accepted at this point. The fluid mosaic model describing how the plasma or the cell membrane is constructed. Now, again, we know this is primarily phospholipids because of their polar nature. The phosphate group is polar, so it's going to align with the water in the intercellular space as well as the high water content of the cytoplasm. So the polar ends of those phospholipids are going to line towards the polar water regions. The fatty acid tails then are going to line up with each other because they're nonpolar, so they want to get together and hang out together. So you've got this bilayer of phospholipids on each side, and that makes up the majority of the mosaic. But you can see there are other different molecules interwoven and put into this membrane. There are cholesterols. Various cholesterols will be embedded within the phospholipid matrix to strengthen and stabilize the bonds between them. And then there will also be various proteins that we're going to talk a little bit about today when we talk about transport of molecules. There are going to be various proteins that allow those cells to move things in and out of the cells. Proteins, ions, different nutrients that the cell needs. So, again, the fluid mosaic model just simply says you've got various molecules that pack together like the tiles of a mosaic. It's a pretty rigid structure. Now, we can force a needle through if we're trying to fertilize, for example, an egg cell and in vitro type of situation. We can actually pierce the egg cell and inject the male nuclear or the male genetic material into that cell. And when you withdraw the needle, the membrane will actually self-seal and form back up. So it is rigid, but it's not like your text talks about. It's not like a balloon where it's going to pop. It will burst. It can't expand infinitely, but they're not. It's not like a layer of plastic or a layer of rubber that you would see in other membranes, like a plastic bag or something like that. So it can self-seal. The molecules can go back together if something pushes its way between them, and something can push its way between them. Other molecules can sometimes pass through the different phospholipids or the other wedge itself in between the different molecules in the mosaic and make its way into the cell. Certain things can, certain things cannot. Polar molecules will group onto the polar surfaces of those phospholipids, but they may or may not be able to get through the non-polar layer of the fatty acids in between. So some molecules need some help. I mentioned there's different proteins embedded within this mosaic layer, and some of those proteins allow for the transport or serve as gates, if you will, through this membrane. That's because this membrane is what we call selectively permeable. Selectively permeable means it's going to allow some substances to pass through. It's going to be selective as to what it allows to pass through. Others cannot. So there's active transport where it requires energy from the cell, the energy, the ATP we talked a little about last time, the cell's currency of energy. Active transport requires some of this energy to be spent, if you will, to move. Passive transport occurs naturally. Naturally occurring phenomena does not require the cell to exert any energy to accomplish the movement. All right, I got to pause there. All right, be back on the next video.